Hi, welcome back to Molecular Inheritance chapter part 2. And in this chapter, we will learn about DNA is the genetic material, and then we also see about the chemistry of nucleic acids. So let's start. So the first one is the major event that took place in the genetic calendar, and we can see that it begins with 1865. Right, when people come across and they say that genes are particulate factors. Then in 71 there was a discovery of nucleic acids, right? And time passed and reaches 1903 when people say that chromosomes are hereditary units. If you remember the certain Bowery experiment, what they said. Then time passed and people say that with new discoveries the genes lies on the chromosomes. Now people know that actually chromosomes are responsible for transferring the gene. Then in 1913, chromosomes are linear arrays of genes with the experiment from on Drosophila. So people were able to understand that these genes are generally arranged in a linear fashion on the chromosomes. Then time passed and we reach 1927, and the mutations are physical changes in genes. So people come to across, the scientists came to know that there's a mutation, and mutations are physical changes that happens in the gene. Then in 1931, so recombination, people discovered about recombination that occurs by crossing over in the that in the meiosis, right, prophase 1, right? And then in 1944, people start experimenting and came to know that DNA is the genetic material. And in 1945 we came to know about the code of protein, that gene codes protein, right? And in 51 protein was sequenced. It was Sanger, right? The Sanger sequenced the protein, and first protein to be sequenced was insulin. Then in 1953, so DNA is the helical structure. DNA has a helical structure. It was proposed by Watson and Crick. You already know that. Right? And time passed in 58. So DNA replicates semi-conservatively. The Stahl's experiment, right? And then in 1961, so genetic code is triplet, right? So people like Crick, Crick postulated then the Nuremberg, and then Govind Khurana. Those people were able to uh, decode that. And there was a Gamow, and Gamow was a physicist who said that definitely there should be a triplet form of nucleotides which should be coding the amino acids. Then in 1977, two things happen. So eukaryotic genes are interrupted. So people found that there are introns are available in the DNA, or introns are available with when the mRNA is synthesized. The mRNA contain coding sequence and non-coding sequence. So those which can be translated into proteins were said to be as exon, and those cannot be are said to be introns. And people discover later that the splicing is a mechanism there through which these introns are removed out and exons are fixed. Then in 1977, DNA can be sequenced. So people understood that DNA is there's a possibility for sequencing of DNA. Again, the Sanger. Sanger discovered the method of sequencing of DNA, and in 1995, bacterial genome sequenced because a small sequence. So since we already knew about the sequencing method, so in 1995, so bacterial genomes were sequenced, and 2000 to 2001 human genome was sequenced. Actually it was proposed to finish by 2000, but it took some time. So anyway, 2001 human genome was sequenced. So you can see that it begins with the history we have. So people like, if you go to Mendel period and see that Mendel said that factors are responsible for transporting the characters or trait from the parents to progeny. And in 2001 we already know that there are sequences in the DNA, which are responsible for those processes. So the whole chapter, if you studying from the beginning, you knew that at the very beginning in genetics we saw that the classical genetics, in which we saw all experiments performed by Mendel. Mendel performed பண்ண எல்லா experiment பார்த்தோம், and that was very 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 basic. அதுக்கப்புறம் கொஞ்சம் வந்து advance போகும்போது we saw that there are genes and there are DNAs which are responsible for transporting these characters. Then That was chromosomal basis of inheritance. Like chromosomes are responsible for transmitting the gene. So basically syllabus has very fantastically designed. Okay, so first you are learning that classical genetics, those experiments were performed by Mendel. Yeah, classical genetics la நம்ம அதை படிக்கிறோம். Right? And when you know that classical disease is a very basic thing, that they say factors are responsible for transmitting of gene. Factors தான் நமக்கு தெரியும், அங்கே ஃபேக்டர்ஸ் தவிர வேறு தெரியாது. மெண்டல் பீரியட் இல்லையா, மெண்டல் பீரியட் வெரி primitive period, and science was very primitive, so you don't know. And then there came a new theory, yeah, chromosomal theory of inheritance. Yeah, and you saw that, you study that chromosomal theory of inheritance, right? So chromosomal theory of inheritance, it came to light after the Sutton and Boveri. They noticed that the chromosomes actually behaves the same way as what Mendel suggested for factors. So chromosome நமக்கு தெரிஞ்சிச்சு இல்லையா. So now we going down the chromosome. இல்லை குரோமோசோம்க்கு உள்ள என்ன இருக்கு நம்ம பார்க்க போறோம். அப்போதான் உங்களுக்கு என்ன வருதுன்னா molecular basis of inheritance or molecular genetics இல்லையா, molecular genetics உள்ள வருது இல்லையா. ஸோ இது நமக்கு தெரியும், ரைட்டா. இதில் போகுது, என்ன போகுது நமக்கு தெரியாது. செகண்ட் ஃபேஸில் வி நோ தட் இஸ் குரோமோசோம், பட் குரோமோசோமில் என்ன நமக்கு தெரியாது இல்லையா. வி நோ தட் குரோமோசோம்ஸ் டூ த ஜாப், பட் வி டோன்ட் நோ வாட்ஸ் குரோமோசோம் இஸ் மேட் அப் ஆஃப், what it contain. So molecular genetics breaks all the information or provide you all the information that how it is in general happens. சரியா சொல்.